Okay, thanks for the kind introduction. Let's jump directly to the intro here. So the way that we raise chicken today is very different from 70 years ago. We used to house chickens in our backyard, and eggs were hatched by the hen. Microbes from the hen and the environment were then introduced to the chicks. However, today broilers are hatched and raised without the presence of their parent flock in commercial barns. Notably, biosecurity measures like chemical disinfectants are also widely used, trying to stop pathogen transmission. With that being said, the chicks have less chance to be exposed to the chicken-derived commensal microbes now. However, early life exposure to commensal microbes are proven to be very important. Studies suggested that the commensal microbes can increase host nutrient utilization and feed efficiency, competitively exclude pathogens, and help program host immune responses, and therefore improve gut health. It is true that we greatly enhance our ability to farm chickens right now. However, pathogens like Clostridium perfringens still haunts the industry. However, foodborne pathogens like Salmonella and Campylobacter are also found in the barns from time to time, as we are now trying to eliminate preventive usage of antibiotics, like mentioned by the previous few speakers. Pathogen control may become more and more challenging in the future. One possible solution may be helping the birds to promote healthy gut microbe host interactions, which may competitively exclude pathogens and prevent the need of antibiotics. Many parts of the world allow reused chicken litters in production. Therefore, many studies looked at recycled litters, hoping that it can help bring back the early life commensal exposure to the chicks. They found that recycled litter has an impact on the chicken early microbiota development, although the effects were not persistent. One research found recycled litter increased the predominance of Faecalibacterium, a butyrate producer in the ceca of both young and mature birds. However, it was also found that the recycled litter may lead to gut inflammation. With that being said, recycled litter may have adverse effect on chicken productivity. I see a Q&A here. Oh, sorry, I answer those questions later. Most importantly, in Canada, broiler producers are not allowed to use recycled litter in production. That makes these studies not applicable here in the North. In practice, water wash and full disinfection are widely used. According to the Chicken Farmers of Canada, chicken barns are required a full sanitation with disinfection at least once a year. We can have the barns water washed between flocks, of course, but many producers still fully disinfect their barns between flocks, hoping to prevent pathogen transmission. But disinfectants may also exclude the good bugs from the previous flock. So to disinfect or not, that is the question. Correct. Currently, limited research is available comparing the effect of these two cleaning methods. Therefore, we want to study how water wash and full disinfection affect flock performance, chicken gut microbiology, and the cecal short-chain fatty acid profile. To achieve this, a big animal trial was performed as a crossover design comparing full disinfection versus water wash treatment between flocks on the production level. We did our study on 7 similarly constructed barns for a total of 4 flocks per barn, that resulting in 14 flocks assigned to each treatment, as we can see from the table. More specifically, for the full sanitation, we try to keep the barns as clean as possible on a production level. In this case, after removal of the used litter, a commercial chemical disinfectant was applied, followed by multiple times of water rinse and fresh litter placement. Notably, the full sanitation takes 2 full days for a crew of 3 people to execute. As for the water washing barns, used litter was removed. The chicken house was then water washed, followed by fresh litter placement. Chickens from both treatments were raised in the same manner by the chicken producers. At day 35 birds from each barn were randomly selected for sampling. Cecal content were collected for gut microbiota analyses, pathogen detection and quantification, as well as short-chain fatty acid analyses. So we hypothesized that the water wash method would impact the chicken gut microbiota that reduced pathogen load. Let's have a look at what we have found. So at day 32, before processing, chicken producers recorded the average body weight and mortality rate. From the 28 flocks we studied, the water wash group showed comparable flock body weight and mortality rate to the full disinfection group, suggesting that the reduced usage of chemical disinfectant does not lead to compromised flock performance. To examine foodborne pathogens, we've enriched Salmonella and Campylobacter from the cecal digesta. Here, occurrence score is defined as the pathogen-positive birds divided by the total birds we sampled in the same barn. During the studies, Salmonella was not detected in either group, indicating that the water wash treatment did not increase Salmonella occurrence in the chicken gut, whereas Campylobacter wise, the water wash group did not show the water wash group did show a significantly lower occurrence score in the water wash group. We have also used 16S RNA sequencing to study the chicken gut microbiota. Both weighted and unweighted unifrac PCOA analyses show that the cleaning method had a modest but significant effect on the chicken gut microbiome, as we can see from the R square and the Adonis P. Whereas alpha diversity, it was not affected by the treatment, suggesting similar microbial species richness and evenness between treatments. The relative abundance of a couple taxa were affected by the cleaning methods. The water wash group showed higher relative abundance of the genus Helicobacter with low abundance of the family Bacillosiae. Helicobacter is a genus that can be found in wild birds. So far, very little was known about its role in the chicken gut. Previous studies found that Helicobacter increased in the chicken gut when polysaccharide hydrolase supplementation was added to their diet, and it's associated with higher mature body weight. Whereas family Bacillosiae-wise, they're spore-forming bacteria that can survive from harsh chemical and physical conditions. So it is not surprising for us to see them being more predominant in the full disinfection group. We have also used the qPCR assay to quantify cecal total bacteria, Campylobacter jejuni, and Clostridium perfringens. In accordance with the Campylobacter enrichment assay, the water wash group showed nearly a log lower copy number of the Campylobacter jejuni hippo gene in the water wash group compared to the full disinfection group. Cecal short-chain fatty acid wise, well, the water wash group showed higher total short-chain fatty acid than the full disinfection group. Specifically, acetate, propionate, and butyrate concentrations in the water wash group were higher in comparison to the full disinfection group, as short-chain fatty acids are important in modulating inflammation and maintaining gut health. Our results suggested that the water wash treatment may be beneficial to the chicken production by giving the birds healthier guts and improved nutrient utilization. Spearmint correlation between short-chain fatty acid concentration and microbes suggested a series of bacteria associating with the outer short-chain fatty acid profile. Notably, Campylobacter is negatively correlated to total short-chain fatty acid, acetate, and butyrate concentration. So we have recently published these data on the AEM. If you're interested, more detail can be found there. And to further understand how the cleaning methods affect the gut microbial functional capacity, we recently sent our cecal samples to do shotgun metagenomic sequencing. The preliminary functional analyses showed that the general microbial community in the water wash group were more abundant in pathway linked to short-chain fatty acid production and amino acid production. When cracked open the functional capacity to the strain level, we found that in accordance with the 16S result, Helicobacter pylori contributed to the initiation of fatty acid synthesis, as well as acetate and lactate production in the water wash group. Although it was proposed to be an opportunistic pathogen, really, there is no concrete evidence showing Helicobacter colonization leads to either human or chicken disease. Therefore, the role of this microorganism in the human and chicken gut needs to be carefully studied in the future. Interestingly, we also found that the water wash treatment Thrive recently discovered obligate anaerobe lesional Clostridium, and revealed that it contributed to a series of complex carbohydrate degradation and short-chain fatty acid production. the colonization of strict anaerobe is often a sign of achieving homeostasis in the ceca. Therefore, it may indicate beneficial effects to the chicken intestinal health. On the other hand, we also found that a pathway linked to lipopolysaccharide synthesis harbored by Campylobacter jejuni were higher in the full disinfection group. In addition, the fatty acid synthesis initiation in the full disinfection group may exerted mainly by E. coli. But what it means regarding intestinal health and immune responses needs to be further studied. So to summarize, there are a few take-home messages. Water wash treatment did not result in compromised flock performance. And at second and day 30, chickens from water wash barns have less abundance of Campylobacter jejuni. And third, barn cleaning methods had a subtle but significant impact on the day 30 chicken microbial composition. And fourth, compared to the full disinfection group, the water wash group had increased total short chain fatty acid in the ceca, and that's supported by the gut microbial functional capacity. All in all, the water wash treatment brought changes to the chicken gut microbiota, which influenced some important microbial abundance and increased the cecal short chain fatty acid concentrations, and that led to a decrease of Campylobacter jejuni abundance in the gut. So in the future, we'll study the relationship between disinfectant usage and antimicrobial resistant genes. Previously, studies on environmental sciences found that chemical disinfectant can trigger ARG mutation and transmission. But in livestock farming today, not much is known about this. And that opens a window for our future study. With the help of shotgun metagenomic sequencing, hopefully soon we'll be able to answer whether the increased usage of disinfectant will affect ARG in the chicken gut. So the project is supported by ALMA, the Alberta Chicken Producers, and the Canadian Poultry Research Council. I would like to thank my supervisor, Dr. Corver and Dr. Willing, for their guidance. Furthermore, our collab partner, Dr. McMullen and Dr. Ingalls, and last but not the least, all my supportive colleagues in the Willing Lab, Corver Lab, and the McMullen Lab. Thank you very much. I'd like to take questions now.
Well, thank you very much for your presentation, Z. There are a few questions in the Q&A section. One relates to one of your first slides regarding the pathogens. Someone was wondering about, does these pathogens can cause disease in birds? And I think it relates to some of your first few slides, since the question appeared quite early during your presentation. So wondering if it's not, doesn't have anything to do with your, yes, probably these pathogens. So I don't know if the person who asked a question, Shana Najimuddin, wants to just elucidate if this is the slide that it relates to. If we're referring to these pathogens, Clostridium perfringens, Salmonella, and Campylobacter.
Yeah, so like it's introduced by other speakers earlier today, Clostridium perfringens is a major headache for necrotic enteritis. Well, it's been decades, and yes, that leads to chicken disease, whereas Campylobacter and Salmonella is mainly concern of them being zoonotic and can transmit to human. And
okay, thank you for this clarification. Another question, are your barns located on the same farm or on different farms? And then how many chickens did you sample and at what age?
So these barns were located in 2 locations in here in Alberta, and they are operated by the same chicken producer. So with the same management, feed and feed supply, they have their regular schedule for management.